Good morning to all and all present here. Today we discuss about the topic is single cell protein. So the single cell protein is derived from cells of microorganisms for yeast, fungi, algae and bacteria to use the grow on various carbon sources for the synthesis of single cell protein. The single cell protein is used for the various animal feeds and human also. First one is, what is single cell protein? Single cell proteins refer to the crude and refined or edible proteins. These are extracted from pure microbial cells or dead cells or dry cells of biomass. They can be used as a protein supplement for both humans and animals. These microorganisms are used for algae, fungi, yeast and bacteria. These microorganisms have a high protein content in their biomass. These microbes can be grown using inexpensive substrates such as agricultural wastes for wood savings, sawdust, corn cobs, etc. These are used for the even human and animal wastes. Sources of single cell proteins. A list of microorganisms used for the production of single cell protein is as follows. First one, fungi, Aspergillus fumigatus, Aspergillus niger, and third one is Rhizobus cyclophene. Yeast single cell proteins are Saccharomyces cerevisiae, Candida tropicals and Candida utilis. Next one is algae. Algae is Spirulina species and Chlorella firinodosa and Contras cereus. And last one is bacteria. Bacteria are Pseudomonas fluorescens, Lactobacillus and Bacillus megatherium. Single cell protein composition are, first one, protein. Fungi is 30 to 45%. Algae single cell protein content for 40 to 60%. Yeast protein content for 45 to 55%. Bacteria protein content for 50 to 65%. Fat content of fungi microorganisms are 2 to 8%. Algae are 7 to 24%. Yeast are 2 to 6%. Bacteria are 1 to 3%. As content of fungi are 9 to 14%, algae are 18 to 10%, yeast are 5 to 10%, bacteria are 3 to 7%. The nucleic acid composition of fungi single cell protein content 7 to 10%, algae content are 3 to 8%, yeast 6 to 12%, bacteria 8 to 12%. Next one, production of single cell protein. The production is carried out the following steps. First one is selection of suitable strain. Second one is fermentation. Third one is harvesting. And fourth one is post-harvest treatment. Last one is SCP processing for food and supply of food. First one, selection of substrate and strain. The strain is otherwise called as microorganisms. First one is in the fungi, bacteria and algae. Microbes are richly grow and selected strains were selected. The rich of protein content and possess suitable growth characteristics. Then the suitable substrates are chosen that are essential for the growth of selected microorganisms. Second one, the selected microorganisms are go to the fermentation. A fermenter is an instrument, so the fermenter carry out the process of fermentation, mainly the mass culture of plant or animal cells. The inoculated medium is then placed in a fermenter when the microorganisms are grow and multiply. And the suitable conditions are given in the fermenter instrument. The temperature, pH and oxygen levels are correctly controlled to optimize the growth of microorganisms. Another one is harvesting. After fermentation, the microbial cells are harvested and separated from the growth medium. However, the harvesting and purification after production of single cell protein production remain a problem. Next one is post-harvest treatment. The harvested microbial cells are then dried to preserve and reduce their volume, making them easier to store and support. Next one, SCP processing for food. The dried cells are chosen and processed to remove impurities and improve their nutritional content and enhance their flavor and texture. So this image represents the desired suitable content for the substrate preparation for agriculture waste and urban wastes and the fermentation process, and then post harvesting method, harvesting method, finally to collect the product of the single cell protein. Next we move to advantage of single cell protein. Large cell single cell protein production has multiple advantages over conventional food production practices such as microorganisms have a high rate of multiplication, which means a larger quantity of biomass can be produced in a comparatively shorter duration. The microbes can be easily genetically modified to vary the amino acid composition. A broad variety of raw materials, including waste material, can be used as a substrate. This also helps in decreasing the number of pollutants. The production is independent of climate conditions. So these all about the single cell protein of microorganisms.